review of Chapter 16. The material that I've given you in Chapter 16 is actually quite brief. The notes are pretty short. Now, DNA is the genetic material. If you look in your book on pages 308 and 309, it reviews the structure of DNA. DNA is a double helix, two nucleotide polymers side by side twisted into a double helix. Each nucleotide, figure 16.5, consists of a sugar, phosphate, and a nitrogen base. The polymers are joined sugar to phosphate, sugar to phosphate, sugar to phosphate, with the nitrogenous bases sticking out. Now in DNA there are four types of nitrogenous bases: T, A, C, and G. Thymine, adenine, cytosine, and guanine. Now on page 309 it shows how the double helix is assembled. The strands of one nucleotide polymer are bound to those of the other according to the base pairing rule. The base pairing rule is A to T and C to G. So everywhere where you see an A on one strand, you're going to see a T on the other and vice versa, and with C and G. Roman numeral 2 in the notes is DNA replication. Now, the best picture for DNA replication is actually in chapter 5. So page 89, it shows you the diagram of DNA, but it also shows you DNA replication. DNA polymerase is the enzyme which replicates DNA. So what DNA polymerase does is it unzips the DNA double helix, exposing the bases. New nucleotides are brought in according to the base pairing rule to assemble a second strand. And then the two new DNA molecules are rezipped. Now, when you have DNA replication, you're going to end up with two new DNA molecules, each with one old strand and one new strand. That's called semi-conservative replication. So the semi-conservative replication model is also shown on page 312, figure 16.10b. Okay, on the same diagram you have A and C. Those are alternative ideas that turned out not to be correct. So it is the semi-conservative model that is the way that DNA is replicated. Now, Roman numeral 3, how DNA is packaged in the chromosomes. View pages 320, 321, figure 16.21. This shows you how the DNA double helix is wrapped by histone proteins into little nucleosomes. The nucleosomes are wadded up into 30 nanometer fibers, and the 30 nanometer fibers are wadded up into chromatin, which is the stuff chromosomes are made of. And that concludes Chapter 16.